Okay, since we in the first half we discussed the amino acid and the protein. In the next half, then we will discuss how the translation, okay, translate those nucleotide sequence into the amino acid, right? So as we know, the gene, so basically I've defined the gene is defined as the order of sequence, all right? Order of sequence of the nucleotide on chromosome that encode a specific functional product, and that specific functional product, okay, mostly is the protein, okay. Of course, that in the last chapter we mentioned some non-coding RNA, but mostly are the protein. The functional product is the protein. And this is just a picture to show you in the eukaryotic cell. So basically, DNA is transcribed to the RNA, and then they will be processed, become the mature messenger RNA. They will be transported out of the nucleus into the cytoplasm, and then bind with the ribosome, and then translate this RNA into the protein, the amino acid, okay, and then form the protein, right? So the next half of the class will discuss this process, right? So the genetic code, basically, the genetic code is written in linear form. We use the ribonucleotide, okay, the ribonucleotide that compose the messenger RNA molecule as a lattice. And then we convert those messenger RNA sequence into the amino acid sequence, okay? And the translation basically will be the conversion of the genetic information in the nucleic acid to the protein. And this is the only event that you will see 3 different RNA work together to translate the genetic information into the protein. So you will see the messenger RNA, transfer RNA, and ribosomal RNA, they all meet together, and then to convert those nucleotide sequence into the peptide. Okay. During this translation process, one very important thing would be how those nucleotide sequence, okay, how those sequences can be translated into amino acid. All right, and so in the early age, then the scientists then they finally developed this the genetic code. All right, so basically each genetic code consists of 3 ribonucleotide lattice. All right, and so we refer this as a triplet code. And so the genetic code is a triplet code, and in which a sequence of the 3 bases is needed to specify 1 amino acid. So the genetic code translates the RNA sequence into the amino acid sequence, okay? And each group of the 3 ribonucleotide, we call it a codon, all right? Like each codon, they specify 1 amino acid. And from this table, you can see that we have, from those 3 bases, because 3 bases, each base, all right? So we have first base, second base, and third base. Each base, you have the four possibility of the nucleotide, right? It's a 4, 4, and 4 here. So for these 4, 4, 4, then you multiply together 4 by 4 by 4. So you have 64 different combination, and this 64 combination will be 64 codons, and these 64 codons will be more than enough to encode only 20 amino acids. Therefore, the genetic code is degenerate, which means more than 1 triplicate. Okay, that means more than 1 codon can encode the same amino acid. All right, so let's say alanine here, for example, alanine, okay, Now be GCU, GCC, GCA, GCG. So you have 4 different codons encode one amino acid, right? That's a degenerate. Okay, so that means more than one codon can encode the same amino acid, and then each amino acid, right, like alanine, each amino acid can have more than one codon, all right? But no codon can encode more than one amino acid. So each amino acid, let's say, for example, alanine, can have 4 different codons. But each of these codons, they can only encode alanine. They cannot encode arginine, all right? So that's what this means. And the genetic code is universal. So that means this code can be used by the virus, prokaryotic cell, archaea, and the eukaryotic cells. Okay, that's the universal. And another feature of the genetic code will be the wobble, the wobble rule, the wobble effect. So basically, the wobble basically allows the third base of a codon to be able to form the non-Watson-Crick base pair with the anti-codon. And that's the reason why you can have more than one codon to encode one amino acid. So as you can see here, GCU, GCC, GCA, GCG, all these 4 codons, they only differ in this third position, right? The third base of this codon. And this third base of the codon are different, but they all code for the alanine, all right? So that means they allow this base pair with the anti-codon, all right? Base pair with the anti-codon, codon will be the non-traditional Watson-Crick base pair, so that they have this wobble effect, so that they can bind and then bring the alanine, all right, when they translate the sequence. Okay, so that's the wobble, and this is just further explanation. The wobble effect, basically, you have multiple codons for single amino acid. They are not randomly distributed, but have one or more bases in common. Okay, so the bases that are common to several codons are usually the first and second bases, right? Like we see here, first and second base, they are usually the same, right? And then they have the room for the variation in the third bases. And so we call the third base will be the wobble base. And the wobble rule indicates the first base, anti-codon U, could recognize either A or G, all right? The base on the anti-codon, the U can recognize A or G. So that means normally U form the base pair with A, but now they can form the base pair with a G. And then C with G, A with U, and G with U or C, right? And so each tRNA, they carry only a single amino acid. So at least 20 different tRNA are needed for the 20 different amino acids. But each tRNA, they carry one anti-codon. Therefore, that means different bases in the anti-codon, they will be able to recognize these different bases for the base pairing. All right, so that's a wobble hypothesis. And the translation of a messenger RNA is the biological polymerization of amino acid into the polypeptide chain. And this process, as I mentioned, needs the messenger RNA, tRNA, and ribosome. Okay, and the ribosome basically is composed of a ribosomal RNA. And the ribosome is the protein synthesizing machine. Okay, for the prokaryotic cell, the ribosome will be 70S ribosome. And the 70S ribosome, we have large subunit, which is 50S, and small subunit, 30S, all right? 30S subunit and 50S subunit. And then 30S subunit is composed of a 16S ribosomal RNA plus 21 proteins. 50S subunit, you have 23 ribosomal RNA and 31 protein and 5S ribosomal RNA. So this is the prokaryote cell. In the eukaryotes, so large subunit is a 60S instead of 50S, and has a 28S ribosomal RNA, all right, 5S ribosomal RNA, 5.8S ribosomal RNA. Small subunit is 40S, all right, and it's 18S ribosomal RNA. So that's the ribosome structure. So each ribosomal subunit, they contain RNA and the protein, all right? So like these large subunit, RNA and protein, RNA and protein. Okay, so that's a ribosome, and the ribosomal RNA, they can have the secondary structure. And this is a typical secondary structure for the 16S ribosomal RNA, okay? 16S ribosomal RNA have 4 general domain, okay, 4 general domain. 1, 2, 3, 4 domains for the 16S ribosomal RNA, and perhaps you have learned a very popular term called NGS, the next generation sequencing. So when people try to use the NGS to determine the like eDNA, like ecological DNA, to determine the bacteria, the presence of the bacteria in the environment, you can use the NGS technology to determine the presence of bacteria and to distinguish what kind of bacteria in the environment or in your gut. And so they use this NGS, and they based on this ribosomal RNA sequence. All right. So as you can see here, we have a V1. You see the V1, variable 1, variable 2, variable 7, variable 8, variable 6, 9. All right. So this variable sequence, this 3 is here, 4 is here, right? So this variable region basically are the region scientists use to determine. They design the probe, the primer for the NGS next generation sequencing, and then the primer. These are the primer sequence to identify each region. For example, use the V3 as the region to distinguish the bacteria composition in the environment. So you can sequence this region, all right, or this region or this region, and so that you will be able to determine the composition of a bacteria, like it's a metagenomic, because you can have the microbiome. So you determine the microbiome in the environment or in the human body. All right. So the 16S ribosomal RNA is a very good signal to determine the different, or to distinguish the bacteria species. And then another player in the translation will be the tRNA, transfer RNA. Transfer RNA is a small RNA that recognize both the messenger RNA and the amino acid, because in the anticodon region, they have these 3 bases. Marina, yes?
I have a question about the last slide. I just thought about it. How come a eukaryotic cell, like in humans, the ribosome unit is 70S versus in the prokaryote, much simpler, it's 16S?
Oh, it's just about the sedimentation coefficient. When they collect the ribosome in the early day, they use the centrifugation to separate the different component, and so they have designed this 16S, 18S. Yeah, it's just the sedimentation coefficient number.
Oh, so it doesn't have to do with anything like involving complexity.
No.
Okay.
No. So for example, the 16S is about 1500 nucleotide long, and then for the eukaryotic cell, that's 18S is about 18 nucleotide long, 1800
Okay,
nucleotide.
I see now.
Yeah, yeah. So the tRNA, all right. So it's the RNA that can form the bridge between the messenger RNA, all right, and the amino acid, all right. And the tRNA interact with the messenger RNA, all right, and also the ribosomal RNA, okay. Because when they form this interaction, anti-codon with codon on messenger RNA, these amino acid in the 3' chain will be processed by the ribosome in the large subunit, the ribosomal RNA, and to form the peptide bond. So this is the bridge between messenger RNA and the ribosomal RNA. And you see the secondary structure is a clover leaf structure. Okay, and 5' is a phosphorylated G, always begin with G, and then 3' always end with CCA, all right, CCA. So we call this acceptor stand, right? The acceptor stand, basically they pair with the 5' end, all right, and also always begin with G as phosphorylated and finish with CCA. And then amino acids bound to here, right? Bound to the 3' hydroxy group of the adenosine, right? That's where the amino acid bind. It's in here. The anti-codon loop, right? You see this anti-codon, right? So this is 5' to 3', 5 to 3. Therefore, the codon will be from here, messenger RNA 5 to 3, like this way, okay? And then we also have these two different loops, and we name this loop after the modified base. So the D loop, T loop, right? This is a T side C loop, all right? T side C loop, and D loop here we have D hydroxyuracil loop, right? So that's the basically the uracil has been modified, yeah. So this is just the structure, secondary structure of tRNA, okay? And then, so as you can see, this is 3'. So this is 5 to 3, and then messenger RNA is 5 to 3 here. So this anti-codon, and this is codon. Codon 1, 2, 3, this position here, now be the first position of anti-codon. So the first position of anti-codon normally follow the wobble effect, like we just discussed, all right? And each tRNA carry only a single amino acid. So at least there are at least 20 different tRNA, because each tRNA will carry a specific amino acid, all right, based on the codon. But in fact, there are about 31 different tRNA, because it's based on the anti-codon and the amino acid, okay? And then to prepare for the translation, all right, to prepare for the translation, okay? So basically, the transcription of DNA does not begin or end at the same place as translation. So we have 5' untranslated region, and also we have 3' untranslated region, okay? Transcription begin at transcription initiation site, depending on the promoter upstream of gene. Translation begin at the start codon and end at the stop codon, okay? So that we have this 5' untranslated region and also 3' untranslated region. Okay. To initiate the synthesis, of course, that we need a specialized tRNA. All right, the tRNA that has been processed, so the tRNA carry the amino acid. All right, for the bacteria. Okay, so for the initiation, they need this modified tRNA. That means that's enformium methionine tRNA. For the eukaryotic cell, just regular methionine tRNA. And the first codon, the initiation codon AUG methionine, normally is removed after the protein is much maturity, right? Then the first codon, the methionine, usually been removed. So the first codon normally not the methionine, but the first one they play as the initiation codon. And for the position of the AUG, how the cell recognize which AUG is star codon, right? So for the bacteria cell, basically that would be the cytokine sequence. Just upstream of the AUG, it's about a 5 to 7 nucleotide upstream of AUG. That will be cytokine sequence, and this sequence match the 3' end of the 16S ribosomal RNA. Okay, it's unique to the bacteria. And then later on, we will discuss a specific recognition rule for the eukaryotic cell. Okay, how they recognize which one is the star codon. Okay, and then to prepare for the translation initiation, you need to have a two event. Two things need to be done before they can start the translation. Number one, you have to activate the tRNA. So activation of tRNA, that means normally tRNA is uncharged. Uncharged, that means the tRNA does not carry any amino acid. In order to make the tRNA to carry the amino acid, then that process is called activation. It's also called charging, all right? The charging of the tRNA. So that means to put the amino acid to the specific amino acid to the specific tRNA. Okay, so that's activation of tRNA. And second event, it would be, remember when we earlier we mentioned the ribosome. We have small subunit and large subunit. So the second event is those two small and large subunit need to be separated, all right? Because the small subunit will need to find the messenger RNA to bind the messenger RNA and then to initiate the translation. So the large subunit must dissociate from the small subunit, okay? So these are the two events need to happen before the initiation for the activation of the tRNA, right? So basically it's like this. So remember the 3' end of tRNA is a CCA, all right, CCA. So the last nucleotide is A, all right, adenine here, all right, and then this adenine need to carry the amino acid. Okay, so adenosine is target for the charging with amino acid, and then the amino acid basically attached by the ester bond between the carboxyl group here, all right, carboxyl group, and this 3' hydroxy group here, all right, the 3'. So we have sugar phosphate nitrogenous base. This is the adenosine, right? And this 3' hydroxy group attach the amino acid. And there are two steps to perform the charging. So the first step is on the top. Second step is here. All right, so the first step would be amino acid plus ATP and then give you amino acid AMP and release the pyrophosphate. So that'll be here, first step. So we have amino acid plus ATP, all right? So you activate the amino acid. So from this, the ATP, you remove this pyrophosphate. You remove the pyrophosphate. remove the pyrophosphate. Okay. And then attach these amino acid, right, to here, right, to this alpha phosphate. So that gives you amino acyl AMP. Okay, that's the first step. Okay, that's first step. So that's the activation of the amino acid here, all right? And then the second step, okay? So first of all, you transfer the amino acid to this AMP here so that you have amino acid AMP and remove the pyrophosphate, okay? Connect this one to here. And the second step, all right? The second step, this is being processed by the amino acid tRNA synthetase. And then the next step, right? So these amino acid AMP, all right? And then you have tRNA, all right? Remember last nucleotide is adenosine, this A here, all right? And then we're going to transfer this amino acid to either 2' hydroxy group or 3' hydroxy group, depending on the class of the amino acid, right? But in general, okay, we just go from this 3' hydroxy group. So transfer this amino acid to the 3' hydroxy group, okay, in the presence of the amino acid tRNA synthetase. So we transfer from here to here, right? And now you have the amino acid tRNA. So this is amino acid tRNA and release the AMP. So you can recycle the TMP. Okay, so that's the first step to activate the tRNA. So let's watch the video here about tRNA charging.
RNA synthetases catalyze the formation of charge transfer RNA. To begin the process, a specific amino acyl tRNA synthetase binds a particular amino acid and also a molecule of ATP at its active site. A covalent bond is formed between AMP and the carboxyl group of the amino acid. A pyrophosphate is released from the enzyme. A specific tRNA that has an anticodon that corresponds to the amino acid binds to the synthetase. The RNA sequence in the anticodon region, as well as other parts of the tRNA molecule, such as the acceptor stem, are important for the recognition between the tRNA and the amino acyl tRNA synthetase. The bond is broken between the amino acid and the AMP, and the AMP is released. At the same time, a covalent bond is formed between the amino acid and the 3' end of the tRNA, which is now called a charged tRNA molecule. The charged tRNA is then released from the enzyme and is now ready to be used in the process of translation on a ribosome.
Okay, that's tRNA charge, all right? And then as I mentioned, there are two different classes of the amino acid. Okay. Okay, class 1 and class 2. So class 1, basically, they first add the amino acid to the 2' hydroxy group, okay? So for the class 1, instead of 3, class 1, they transfer this amino acid to the 2' hydroxy, and then they transfer to the 3' hydroxy. Okay, that's class 1. And class 2, they just transfer directly to the 3' hydroxy. Okay, so that's the class 1, class 2, depend on the different. This is class 1, we have 10 amino acid belong to the class 1, like arginine, cysteine, and class 2 alanine. All right. So this is details. So the first step, okay, we charge the amino acid. Okay, so we have ATP amino acid, right? And then you connect this amino acid to the AMP, all right? So we have AMP adenine, and then amino acid release the pyrophosphate. And then the class 1 amino acid tRNA synthetase basically would transfer this amino acid, all right? All right, this amino acid to the 2' hydroxy here, okay? To the 2' hydroxy here. and then they have extra step, and then to move to the 3' hydroxy. In the end, everything has to be in 3' hydroxy, okay? And then the class 2 basically just go all the way down here. That's class 2. And also, as we know that for the tRNA, those tRNA synthetase to function those amino acid and the tRNA, all right, the synthetase, this enzyme, they try to match the amino acid and tRNA, all right? And then for those, because in the pool of a cell, so many different things. So that's why some people propose a second genetic code. The second genetic code, I mean, in the tRNA, all right? tRNA, they have several different sequence features that will help those synthetase to recognize. And so they don't need to read the anti-codon sequence. So they know what kind of the amino acid they should bring to the specific tRNA. And that's called the secondary, second genetic code. Okay, so basically it will be a set of sequence elements that can be recognized by specific amino acid tRNA synthetase. And these sequence elements, including, for example, at least one base in the anti-codon, all right? One base in the anti-codon, okay? One or more of the 3 bases pair in the accepted stem, all right? And also unpaired base preceding to the CCA, the unpaired base here, all right? So these are all sequence elements, all right, that can be the second genetic code, so that the tRNA synthetase can recognize tRNA immediately and bring the specific amino acid to that tRNA. Okay, that's a second genetic code. And of course, there's also a proofreading mechanism just to make sure the amino acid tRNA synthetase bring the correct the amino acid to the tRNA. Okay. So the two mechanisms. The first one, the amino acid tRNA synthetase, make an intricate series of contact with both the amino acid and tRNA in the enzyme active site to ensure the most part that only the correct substrate are selected from the large cellular pool of a similar candidate. And second, that will be the enzyme possess a variety of proofreading activity. Okay, so they can serve to hydrolyze the mismatched amino acid before or after transfer to the tRNA. Okay, so these are two mechanisms to ensure the correct amino acid is attached to the tRNA. Okay, so that's the first event to activate the tRNA, to charge the tRNA with amino acid. And then the second event will be a dissociation of ribosome. Okay, so dissociation ribosome, basically you need to separate 30S and 50S subunit, and that will require, okay, the initiation factor 3, IF3, okay, because IF3 bind to the free 30S subunit and prevent the reassociation. Okay, that's the important factor, initiation factor 3. And then of course the RF and EFG, these two proteins, they can promote this dissociation. So here, that's for the protein synthesis in the beginning, all right? You need to form the 30S initiation complex, all right? 30S initiation complex, as I mentioned. First of all, tRNA need to be charged, and number two, the 30S need to be dissociated from the 50S. So now you have free 30S, and it's bound by the initiation factor 3, all right, to prevent the reassociation, and also by the help of IF1 initiation factor 1, all right, and also we have initiation factor 2, okay. And during the initiation stage, all right, first of all, the 16S ribosomal RNA in this small subunit, okay, will bind, recognize the cytogamy sequence, will bind the cytogamy sequence. So Now bring this to here. So they bind together, all right? They bind together. The IF3 bind the 30S, but the IF1 and IF2 they can stabilize the binding. Okay. And once they bind together, all right, the IF2 will be released. 1 and 3 will be also released, all right? And also this process, they need a GTP, all right? GTP is here because the GTP hydrolyze the GTP, the hydrolysis of GTP release the energy to help the release of the initiation factor and also the binding of large subunit to the small subunit, okay? And then at this point, as you can see that the tRNA already bind to the star codon, right? The formylmethionine already bind to the star codon, and then they join the large subunit, okay? And the binding of the messenger RNA to the 30S ribosomal RNA, basically, it's done by the 16S ribosomal RNA, all right? 16S ribosomal RNA, so they can recognize the shotgun sequence, all right? And this is, see here, this is the messenger RNA, and here you GGA, GGU, the shotgun sequence. That's complementary to the 3' end of the 16S ribosomal RNA, okay? And then we know this is the AUG. So there's a 7 nucleotide in between the shotgun sequence and the star codon AUG. Okay. And then the IF2 basically is a major factor promoting the binding of the formylmethionine tRNA to the 30S initiation complex. Okay. And the GTP is required for IF2 binding. All right. And the GTP is hydrolyzed after 50S subunit join the 30S subunit and then form the 70S initiation complex. Okay. And then once this is done, that's translation initiation. Okay. And then the elongation basically begin when the second amino acid is joined. All right. So for the bacteria cell, okay, the formylmethionine tRNA bind to these sites called P sites. There are 3 sites in the large subunit. We have EPA, all right? So the star codon begin from the P site here, all right? And then this is translation initiation in order to move into the elongation, okay? The A site will need to have the incoming amino acid tRNA, which will be the second amino acid tRNA. They need to bind to the A site in this. So here, all these factors join to help the second tRNA, amino acid tRNA to bind to the A site. So this will start the translation elongation. Basically, the triplicate in the A site, they need to match the anticodon of the incoming amino acid tRNA, and we need a GTP and the 3 elongation factor like EFP, all right, and the EFTU, and EFTS. These 3 elongation factor to help, they are required. So basically EFTU, all right, basically EFTU will guide the amino acid tRNA, all right, into the part of the A site, right? The EFTU will help the amino acid tRNA, okay, to bind to the A site, okay? And the EFP and GTP, basically GTP provide energy, right, to dissociate the EFTU. And EFP is bound adjacent to the P site, right? Adjacent to P site and the E site. and that can help the formation of the peptide bond. Okay, that's the EFP. And the EFT basically help the regeneration of the EFTU and the GTP. Okay, that's the EFTS. Okay, so basically the EFTU will help this amino acid tRNA, all right, to bind to the A site in the presence of GTP, and then they release EFTU, and then also the hydrolyze the GTP, release the energy, right? And then the EFTS help the EFTU, right? And the GTP together, all right? They can help these together to form these complex again together. And then they form these peptides through the EFP, the help of EFP, okay? And we have this peptidyl transferase to help to form the peptide bond. And then the amino acid can be transferred to the next one, okay? To the amino acid tRNA at the A site, okay? From the P be transferred to the A site, okay? Then it'll become a dipeptidyl tRNA. And then this tRNA will become empty, no amino acid attached at the P site. And then these uncharged tRNA will move from the P side to the E side, okay, to the E side. And then the peptidyl tRNA move to the A side, all right? And this one, once this empty uncharged tRNA move to the E side, they will be released. And so they will be recycled to charge again, so they can come back, all right? And now this one, the peptide tRNA will go to the P side, and then incoming amino acid tRNA will recognize the codon and bind, so they will repeat this cycle for the elongation, okay? For the elongation. So basically, during this process, the 30S subunit will be the decoding center, because they provide a messenger RNA and also provide the interaction between the codon and anti-codon, all right? And then the large subunit basically will be the place for the peptide formation to form the polypeptide. And then in prokaryote cell, translation begin very soon after the messenger RNA transcription. So that basically the gene is being simultaneously transcribed and translated. So we call this as a coupled translation. Okay, in this situation, several molecules of RNA polymerase are attached to a single gene. And then you have several molecules of a messenger RNA are transcribed, and each messenger RNA molecule is still being transcribed. Then a number of ribosomes already attached to the messenger RNA, and then they start a translation. And then the termination. So for the termination, basically, is signaled by one of the three nonsense or termination codon, okay, which are not charged with the amino acid, okay. And once that they come to the these termination codon, then release. We have the release factor, okay. Release factor basically will bind to the stop codon, all right. Stop codon basically RF1, that's release factor 1, recognize UAA, UAG. RF2 recognize UAA, UGA, okay. And then RF3 basically facilitate the binding of RF1 and RF2 to the ribosome. So that's the termination. And then for the eukaryotic cell, only two are release factor. We have the ERF1 and the ERF3. Okay, ERF1 recognize all three termination codon, and the ERF3 basically help the ERF1 to release the finished polypeptide. That's the termination. Okay, so let's watch this video.
is the process whereby the base sequence of a messenger RNA is converted into the amino acid sequence of a polypeptide. This process involves an mRNA, tRNAs, amino acids, a ribosome, and other accessory.
So here will be the small subunit, and here the big one, large subunit. And small subunit, 30S ribosomal RNA, large subunit, 30S subunit, 50S subunit. Then we have 16S ribosomal RNA in small subunit, 23 ribosomal RNA in large subunit. Large subunit, we have E, P, A, 3 different sites, okay? And then they begin from the formula methionine tRNA bind to the P site, okay?
Proteins. mRNA consists of three main parts. The portion from the 5' end to the translation start codon is the 5' untranslated leader region, more simply called the leader. The central portion is called the reading frame. It commences with the start codon, AUG, followed by a series of 3 nucleotide codons, each of which specifies an amino acid, and it ends with one of the stop codons, UAA, UAG, or UGA. The final portion of the mRNA consists of the section from the stop codon to the 3' end, called the 3' untranslated trailer sequence, more simply called the trailer. mRNAs vary in length because of differences in all 3 parts of this molecule. Transfer RNA functions to bring amino acids to the growing polypeptide during translation. It consists of a single RNA chain of about 75 to 90 nucleotides, each tRNA having a different sequence of bases. After it is transcribed, the tRNA folds up into a characteristic cloverleaf structure. The anticodon contains a 3 nucleotide sequence that is complementary to a codon on the mRNA. The amino acid is attached to the 3' free end top of the cloverleaf. The ribosome assembles amino acids into a polypeptide. Ribosomes consist of 2 parts, a large subunit and a small subunit. Ribosomes are made of ribosomal RNA and various proteins. The active ribosome contains 3 tRNA binding sites, namely E, exit, P, peptidyl, and A, aminoacyl. In prokaryotes, translation initiation occurs in 3 steps, as follows. First, the small ribosomal subunit, 30S subunit, complexes with 3 proteins named initiation factor 1, initiation factor 2, and initiation factor 3. A molecule of GTP also attaches. The ribosome IF-GTP complex recognizes a sequence in the leader, upstream of the AUG start codon. This sequence is called the ribosome binding site, or the Shine-Dalgarno sequence, after its discoverers. This sequence is complementary to a sequence at the 3' end of the 16S ribosomal RNA of the 30S ribosomal subunit, and serves to align the ribosome on the mRNA in its proper reading frame. Second, translation begins at the start codon, AUG, a few bases downstream of the ribosome binding site. The first AUG in proteins specifies a modified form of methionine. This modification consists of the addition of a formyl group to methionine, making it formylmethionine. The anticodon of this initiator tRNA containing FMET complements AUG. Upon binding of FMET tRNA to mRNA, IF3 is released, with the remaining assembled components called the 30S initiation complex. Lastly, the large ribosomal subunit, 50S subunit, binds to the 30S initiation complex. GTP is hydrolyzed, and IF1 and IF2 are released. The final complex is called the 70S initiation complex. The FMET tRNA is now situated in the P site of the ribosome, with its anticodon hydrogen bonded to the mRNA start codon. Initiation is now complete, and elongation follows. Once translation initiation is complete, elongation begins. The elongation phase consists of 3 steps: binding of a tRNA to the ribosome, formation of a peptide bond between adjacent amino acids, and movement of the ribosome to the next codon. Elongation continues 1 codon at a time until a stop codon is reached. As initiation concluded, F-met tRNA was hydrogen bonded to AUG in the P site of the ribosome. The next codon is in the A site. In this case, the codon is UCC, which encodes serine. The serine-bearing tRNA in the cytoplasm forms a complex with elongation factor TU and GTP. This ser-nil tRNA EF TU GTP complex enters the A site where the tRNA anticodon forms hydrogen bonds with the UCC codon. Upon binding, the GTP is hydrolyzed and EFTU is released and recycled. With a tRNA in both the P and A sites, a peptide bond can form between adjacent amino acids. This reaction is catalyzed by peptidyl transferase residing in the large ribosomal subunit. The first step involves breaking the bond between F-met and its tRNA in the P site. Next, a peptide bond forms between the now freed F-met and the serine, which is attached to its tRNA in the A site. Peptide bond formation is a condensation reaction that results in the release of water. At one time, it was thought that peptidyl transferase was an enzymatic protein. However, more recently, it was discovered that peptidyl transferase is a catalytic RNA molecule. An RNA capable of catalyzing a reaction is called a ribozyme. Once the peptide bond has formed, there remains an uncharged tRNA in the P site and a tRNA with a growing polypeptide chain in the A site. The ribosome now moves 1 codon along the mRNA with the help of EFG and another GTP. An EFG-GTP complex binds to the ribosome, hydrolysis of GTP occurs, and translocation begins. The uncharged tRNA moves from the P site into the E site, blocking the next aminoacyl tRNA from attaching to the A site until translocation is complete. The tRNA in the A site, still bound to its codon, now moves into the P site. Once this tRNA is bound correctly in the P site, the uncharged tRNA is ejected from the E site. The A site is now open for the next aminoacyl tRNA to enter, allowing the elongation process to continue. An average polypeptide consists of about 300 amino acids. Let's watch the elongation process once again.
So that's the elongation. And then termination.
Elongation continues until the ribosome reaches one of 3 stop codons, UAG, UAA, and UGA. Unlike other codons, stop codons do not code for any amino acid, so no normal tRNAs have anticodons that complement these stop codons. The ribosome recognizes polypeptide chain termination with the help of proteins called release factors. RFs read the stop codon and initiate a series of specific termination events. E. coli has 3 RFs, RF1, RF2, and RF3. RF1 recognizes UAA and UAG, while RF2 recognizes UAA and UGA. RF3 does not recognize any stop codons, but stimulates the subsequent termination events. Elongation and ribosome translocation result in a stop codon entering the vacant A site. RF1 or RF2 binds to the stop codon, which triggers peptidyl transferase to cleave the now complete polypeptide from its tRNA located located in the P site of the ribosome. The tRNA is then displaced from the P site of the ribosome, followed by disengagement of the RF and the 70S ribosome from the mRNA, and separation of the ribosome into individual 30S and 50S subunits.
So now we just watched all three videos from translation, initiation, elongation to the termination. Okay.
Elongation.
So that would be the prokaryotic translation. And then for the eukaryotic translation, okay, so remember in bacteria, begin with the N-formyl methionine. For the eukaryotic, that's a regular methionine, okay? It needs a cytokine sequence to show the ribosome where to start, okay? But the eukaryotic, there's no cytokine, but there's some other signal, okay, that can help the methionine to come to the start codon. And also another difference is for the prokaryotic cell, you have the E site, P site, and A site. For the eukaryotic cell, right? Eukaryotic cell, only two sites, only the peptide transfer site and this amino tRNA site here. P site and A site, only two sites, all right? Only two sites. And then for prokaryotic cell, 70S, it's 80S, 30, 50, 40, 60, all right? And then this is just some hypothesis for the ribosome to identify the start codon, all right? So basically, they locate the start codon by binding to the 5' cap because the messenger RNA is capped, all right? The methionine, the methylated guanine, right? So they bind here, right? Bind the 5' cap, okay? And then they scan downstream to find what they bind this cap, and then scan the downstream to find the first AUG, right? To find the first AUG in the favorable context, and the favorable context will be A. Right here, AUG was +1, and then purine in the -3, CC here, -1, -2, -3. It's either A or G, that's purine, okay? Either A or G. And then there's G in the fourth position, +4 position. So this rule, okay, can help the ribosome to identify the AUG, okay, the start codon, all right? Remember, -3 will be the purine. So purine will be either A or G, and number +4 must be G, okay? So that's how they identify the start codon for eukaryotic cell. And then for the initiation, all right, so basically you have to assemble the 43S pre-initiation complex. The 43S initiation complex, basically you have the tRNA the amino tRNA that carry the methionine, right, which is met tRNA. Then you also need a 40S subunit, GTP, and eukaryotic initiation factor. So that will be the EIF1A, EIF2, and the EIF3, all right? So we need to, first of all, 80S need to dissociate, right? The 60S dissociate from 40S, then 40S, then bind with the EIF1A, EIF3, okay? Then 43S, you also have this methionine tRNA, EIF2 all come together. Then you form this pre-initiation complex, okay? And then the step 2, the messenger RNA, all right? Messenger RNA is recruited, all right? The 5' cap here, all right? Basically, the 5' cap will orient the ribosome to the correct AUG. So they will bind to the correct AUG through the scanning mechanism. Then the EIF4A, okay, EIF4A, EIF4B, EIF4E, and the EIF4G, they can help the 43S pre-initiation complex bind to the messenger RNA. Okay, so now we have 40S subunit, we have messenger RNA, we also have 7 EIF, okay, the eukaryotic initiation factor, okay, that constitute the 46 48S. So that the whole thing now become the 48 as the pre-initiation complex, and then you move downstream, okay, until it encounter the first AUG, all right, and then once they encounter the AUG, then the 60S ribosome is recruited, and then they form the ATS initiation complex. Then the GTP is hydrolyzed, then all the initiation factors are released. So that would be the eukaryotic initiation of translation. And this is just the different function of the eukaryotic initiation factor. Okay, so EIF4 basically is a cap binding protein that will bring the 40S subunit to bind the 5' cap of messenger, and that's EIF4. And then the elongation in the eukaryotic cell is very similar to the prokaryotic cell. Basically, you have the same mechanism of peptide transferase and also the ribosome translocation. Okay, the only difference is in the eukaryotic cell, they don't have the E site, only a P and the A site. Okay, and there are two eukaryotic elongation factors. So we have EF1, EF2, right? And then termination basically the same. When the ribosome encounters the stop codon, then we also have the release factor to bind to the stop codon and catalyze the hydrolysis of a bond between the C-terminal amino acid and tRNA and release the peptide. Okay, that's termination. So and then some post-translational modification, okay, for protein. So basically, as I mentioned earlier, so the protein, they need to go through a process so they can become a functional protein. And so that process, so it's a modification, right? And it has to go through this molecular sharpening to help the newly made polypeptide to fold properly, so they will not become misfolded protein, and they will become the functional protein. Okay, molecular sharpening. And so that's molecular sharpening. And in E. coli, there's another special sharpening. It's called a trigger factor. The trigger factor basically associates the large ribosomal subunit and catch the newly synthesized polypeptide. All right. And then the hydrophobic region of the nascent polypeptide are protected from the inappropriate association until the appropriate subunit is available, so they can form the correct functional protein conformation. And for the protein degradation, okay, so the protein degradation, okay, because the protein are in dynamic state, and so the turnover is very quick, all right, and then protein degradation basically would be a hazard to the cellular process, and to control the hazard, the degradation pathway basically need to be restricted to a degradative subcellular organelle like a lysosome, all right, or proteasome. And then for this reason, the regulation protein, all right, through the degradation is an essential cellular mechanism, all right. And in eukaryotic cell, the most common degradation mechanism in proteasome is by the ubiquitination, okay. Ubiquitination is a protected mechanism, okay, ubiquitination. Basically, ubiquitin, all right, is a small polypeptide, and it's highly conserved in the eukaryotic cell. When the ubiquitin is linked to a protein, then they can bring the protein to the proteasome, and then the protein can be degraded in the proteasome. Okay, so the ubiquitin bind to the protein and bring the protein to the proteasome for degradation. Okay, and that involves several steps. Okay, and 3 different proteins. So E1, E2, and E3. E1 is a ubiquitin activating enzyme. Okay, E1, the ubiquitin activating enzyme, basically become attached through a ester bond to the C terminal of the glycine residue of the ubiquitin. And then the ubiquitin is transferred from E1 to the SH2 group of E2. And E2 will be the ubiquitin carrier protein. So E1 is a ubiquitin activating enzyme, all right? So E1, all right? And then you transfer the ubiquitin, okay, to the E1, like here, all right? The E1 and then ubiquitin. And then E2 is the ubiquitin carrier protein, all right? So E1 transfer the ubiquitin to the E2. So E1 transfer the ubiquitin to the E2, all right, like this E2, all right? And then in protein degradation, the E2 ubiquitin transfer ubiquitin to the free amino group on the protein selected by the E3, all right, which is the ubiquitin protein ligase, that's E3. So we transfer the, so the E3 is the ubiquitin protein ligase, okay? Basically, E3 bind to the protein, so we have E3 protein, all right, and E2 with ubiquitin. E2 is the ubiquitin carrier protein, all right, and then this E2 transfer this ubiquitin to this protein, all right, see, and now release E2 and E3, so protein will carry the ubiquitin. So that will become the ubiquitin linker protein. And then I would direct the protein to the proteasome for the degradation. So that's the process for the protein degradation, okay? And then, of course, there are also some protein, although the protein ubiquitination is the signal for degradation, and also it can have some non-degradative function. Okay, so for example, some small ubiquitin-like protein modifier, okay, we call the SUMO, small ubiquitin-like modifier, protein modifier, okay, that's SUMO protein. So basically, it's a highly conserved protein. They covalently ligate to the target protein, okay, through the 3 enzyme conjugating system. And then when they attach to a protein, then the protein, they can participate in several important cellular functions like a DNA repair, transcription, protein homeostasis, protein trafficking, okay, all kind of the important cellular function. So basically the SUMO, before the conjugation, the SUMO here, all right, they need to be proteolytically processed. All right, so that means they have to cleave the protein. All right, cleave the protein. They cleave the protein, they remove the C terminal of the glycine, this glycine motif. So they remove this terminal here, right? C terminal of this motif. So now you remove this C terminal. So now it becomes SUMO with glycine motif, all right? Then this SUMO then are activated by the E1 enzyme, okay? They'll be activated by the E1 enzyme. Okay, E1 enzyme. This is ATP dependent reaction, so that the SUMO can is transferred to the catalytic cysteine. Okay, catalytic cysteine residue of the E2 enzyme. All right, this is E2 enzyme, so this is E1. You connect this here and then transfer to the E2, the cysteine. All right, residue here, and then finally you have E3, bring the protein, the target protein, so that transfer the SUMO to this target protein through this lysine residue, and then bring the protein to the specific location for the specific cellular function. Okay, that's a SUMOylation, and this is ubiquitination. This is for protein degradation. This is protein for specific function. All right. Then we will watch the last video.
Options are typically thought of as small chemical groups that are attached to protein side chains. Yet one class of modification involves the covalent attachment of an entire protein to lysine side chains on a target protein. The most common of these proteins is ubiquitin, a compact 76 amino acid protein found in all eukaryotes. Ubiquitin is covalently attached to lysine side chains in a process known as ubiquitination, also called ubiquitylation. In this process, the C-terminus of ubiquitin is covalently attached to the lysine epsilon amine by a peptide-like bond known as an isopeptide linkage. This modification adds about 8,500 Daltons for each ubiquitin attached, which is more than 100 times the molecular weight of a single phosphate group. A substrate protein can be modified by the attachment of one or more ubiquitin monomers or any of the different types of polyubiquitin chains. The polyubiquitin chains can occur because ubiquitin itself has 7 lysine residues, each of which can be ubiquitinated. Ubiquitin can also be attached to the N-terminal amine of another ubiquitin, resulting in linear polymer ubiquitin proteins. There are thus 8 different types of polyubiquitin chains, each distinguished by the ubiquitin lysine through which one ubiquitin is attached to the next. Each type of ubiquitin modification, whether it's a single ubiquitin or one of the different types of chains, has a distinct biological function. Perhaps the best understood is the targeting of proteins for degradation by covalent attachment of a K48-linked polyubiquitin chain. The K48-linked chain is recognized by the proteasome, which proteolyzes the protein into short peptides. In contrast, modification with K63-linked polyubiquitin chains modulates the activities of proteins with critical roles in the DNA damage response and in the inflammatory response. Let us now examine the cascade of reactions that lead to ubiquitin attachment to a protein. These reactions are catalyzed by 3 different enzymes known generically as E1, E2, and E3. In the first step of this reaction, the ubiquitin C-terminus is covalently attached by a thioester bond to the active site cysteine of the E1 or ubiquitin activating enzyme. This reaction is powered by ATP hydrolysis. The E1 ubiquitin conjugate then binds to an E2 or ubiquitin conjugating enzyme and catalyzes the transfer of the ubiquitin onto the E2 active site cysteine. This step is a transthiolation reaction, since the final product is also a thioester-linked ubiquitin. In the final step, the ubiquitin is transferred from the E2 to a lysine on the target protein. This step is orchestrated by the E3 ligase, which brings together the ubiquitin-loaded E2 enzyme and the substrate, and helps catalyze the transfer of ubiquitin from the active site cysteine of the E2 to the amino group of a lysine side chain, forming a covalent bond. Multiple classes of E3 ligases catalyze this last ubiquitin transfer step in somewhat different ways. The most common type are the ring E3 ligases, which contain a so-called ring domain. This domain binds both the E2 ubiquitin conjugate and the substrate, and stimulates the direct attack of the substrate lysine on the E2 ubiquitin thioester. This attack leads to the covalent attachment of the ubiquitin C-terminus to the lysine. Another E class, such as the HECT ligases, contain an active site cysteine. In this case, the HECT E3 binds the E2 ubiquitin conjugate and the substrate, but first stimulates transfer of ubiquitin from the E2 to the active site cysteine of the E3 in yet another transviolation reaction. The HECT ligase then catalyzes attack of the substrate lysine on the ubiquitin E3 thioester, and the ubiquitin C terminus is conjugated to the substrate lysine. Polyubiquitin chains consisting of one ubiquitin C terminus linked to a lysine residue, or N-terminal amine, are assembled in the same E1, E2, E3 enzymatic cascade. For example, K48-linked polyubiquitin chains are formed by linking the C terminus of one ubiquitin to lysine 48 in the next. This linkage specificity is thought to be governed primarily by the E2 enzyme. Although most eukaryotic cells contain a single type of E1 enzyme, they typically have a dozen or more different E2 enzymes and hundreds of E3 enzymes. For example, the human genome contains around 30 E2 enzymes and at least 500 E3 enzymes. The large number of E3 enzymes and the great diversity in the different domains they contain, in addition to E2 binding regions, reflects the need of the cell to very specifically target particular protein substrates for ubiquitination, as well as its need to interact with a variety of E2 enzymes.
Okay, so that's the ubiquitination. So that's the end of the chapter 4.